Hello and welcome to the Point Blank Show. My name is Abhishek. I'm honored to be joined by Dr. Thomas Cech, who shared the Nobel Prize in Chemistry with Sidney Altman back in 1989 for the discovery of the catalytic properties of RNA. Their work demonstrated that RNA is not only a molecule that carries genetic information, but can also act as a catalyst in biochemical reactions. That was a role that was previously thought to be exclusive to proteins. This has helped us understand a range of subjects like the mysteries of the origin of life, mRNA vaccines, among many other things. Tom's book titled The Catalyst, RNA and the Quest to Unlock Life's Deepest Secrets was released recently and is a fascinating read. Thank you so much, Tom, for doing this.
I'm glad to be here, Abhishek.
Thank you. And first of all, full disclosure, I'm not even going to pretend that I'm remotely qualified to speak about this subject with you. And the irony is not lost on me. This is a commerce graduate with a background in management talking chemistry to a Nobel laureate. So the only thing connecting me to science is my mom, who recently retired as a science teacher, school science teacher. She would teach chemistry there, but much to her chagrin, I didn't take up that subject. What are your first recollections of science as a kid? What introduced you to this world?
Abhishek, I knew that I wanted to be a scientist from the time I was perhaps 5 years old, my earliest memories. I was always intrigued about how nature worked. I was very interested in rocks and minerals and fossils, and I would collect them. I would hit them with a hammer to see what was inside. And I had a physician father who was sort of driven into medicine by his family for financial reasons, but always really wanted to be a physicist. And so he would talk on any family excursion, he would always talk about nature from a very scientific viewpoint. And this resonated with me. And so I always knew I wanted to be a scientist.
And I've read somewhere that you were also a fan of sitting outside and looking at meteor showers and trace the paths of meteors in the sky, somewhere in middle school, perhaps. Is that what it was?
Yes, I would set up my old camera and I would, on a time exposure so that I could capture the meteorites blazing across the sky. Astronomy was very exciting to me when I was in middle school, and then later I turned more to physics and then to chemistry, very last to biology.
And how did your professors back in the day react? How do they react to curious students? I'm imagining that you were terribly curious back in the day as well, knocking on professors' doors to know about things that other students may not ask.
Yes, I lived in a university town called Iowa City, Iowa, and that's where the University of Iowa was located. And I had even though I am a rather shy person, I was so excited about the science that I would knock on the doors of the geology professors. And quite remarkably, they were very kind to me, and they would call me into their office and tell me about some things that they were doing. And I hope that I have that much patience with young students today.
And talking about patience, you've confessed in the past that you are not, patience doesn't come very naturally to you, or didn't when you were in your 20s. And lab work, which is pretty much where all the discoveries take place, require a lot of patience. How do you square with that? Or have you changed over time?
Yes, that impatience, as you called it, can be a useful quality, but it drove me out of chemistry where the experiments tended to be very long, take a long time to do. And I much preferred applying my chemical knowledge to biological systems where you can often do an experiment each day. And already the next day you get an answer. Sometimes the answer is that you made a mistake and it didn't work, but there's constant feedback between experiment and idea. And for me, that was very attractive.
And then came a day when you called yourself a DNA guy in slang. Tell us about it. How did you get attracted towards the field itself and you decided to dedicate your life to it?
Yes, when I became disillusioned with chemistry, I was in the uncomfortable position of being a chemistry graduate student at the University of California in Berkeley, and knowing that I really didn't want to do chemistry. And so I was fortunate to find a professor, his name was John Hurst, who had been trained as a physical chemist at Caltech, but now was working on chromosomes and DNA, and I didn't really know what this was, but I knew that it was very different. I mean, I knew a little bit about it, but I didn't know it in any depth. And I knew that it was very different from studying small molecules hitting into each other in the gas phase. And so maybe this would be something that would be of more interest to me. And as soon as I joined his laboratory and started doing the experiments, I found that this was really what I loved to do. And DNA, of course, being the master molecule responsible for all of life, the storehouse of genetic information, who could possibly want to study anything more important than DNA?
Right. And I think for non-science background folks like me, the word DNA, I mean, it's a verb. We've used it. Let's say the brand should have a particular DNA. The organization's DNA is this. So something that is very fundamental and hardwired into the individual or the company is supposedly that. And do you see RNA becoming a verb someday? Do you- do-
I really was not very interested in RNA when I was a student. I thought it was just the product of the DNA. And, the DNA was the mastermind, and the RNA was just a servant working for the DNA. So it took some time for me to wake up to the fact that RNA had many wondrous qualities, actually much more than DNA.
In one podcast, I remember you compared RNA. You said it was more like a chorus or a background singer to the pop diva that DNA was, and it's like for me, it's a stuntman versus the main actor is how people perceived it. So very briefly, what does RNA do, which you realized later, as you suggest, should get the necessary spotlight? What are some of the functions of RNA which blew your mind back in the day?
So at the time, we really didn't know about most of these functions. We knew that RNA could store information, and in fact, many viruses, such as the SARS-CoV-2, but also Ebola virus, the influenza virus, these don't even have DNA. They just use RNA as their genetic information. And we knew that RNA could be a messenger, and that role has been turned into vaccines with the mRNA vaccines. But the really wonders of RNA to me are all of the things that it can do beyond being a coding molecule or a message. And these include being able to make cells immortal, being able to drive CRISPR genome editing, which I think many people have heard about, and to act as a biocatalyst and perhaps provide the secret of how life originated on the planet Earth. So these non-coding functions of RNA is what gets me really excited.
That last bit, which you very casually mentioned about the life, how life began, could you even try and how does one wrap one's head around it? How do you recreate what happened billions of years ago? Because you obviously didn't have cameras or anything remotely as close to that. So, how do you even go about approaching such problems, Tom?
You are absolutely right, Abhishek. Anyone who says that they have proven how life started on the planet or that they know how life started, they are exaggerating. We can come up with laboratory experiments that tell us what is plausible, what might be realistic, but it is difficult to know exactly what happened. But here's the story. So for many years, scientists were puzzling over how the first reproducing system, very primitive life could have gotten started on the planet Earth. Now, if you think about what you need for life, you need to have an informational molecule, right? You need to have some information that you pass down to the next generation. Well, we know now, for us, that is DNA. And also for all the plants and all of the bacteria and every life form on this planet, it is DNA. But DNA doesn't do anything by itself. It just sits there. It needs to have an enzyme, a protein catalyst to make one mother copy into two daughter copies, right? So you need some machinery to reproduce it. So how could this possibly get started then? If you need in the same little place at the same time, both the informational molecule and a catalyst to copy it by some random chemical processes, seems very difficult. So now, following on from the discovery of RNA catalysis, we know that RNA, in addition to being an informational molecule, can be a copying machine. Think about a photocopier in your office, right? The RNA can both be the thing that is being copied, but it can also at the same time be the machine that is doing the copying. So this is a potentially shortcut to understanding how life could have gotten started. We call this the RNA world hypothesis, that at the beginning there was RNA replicating itself, and that the proteins and the DNA came later.
And if you can replicate this in the lab, then the odds are that that may have happened back in the day.
Yeah, that's, some people think that they just understand that must have been what happened, that RNA must have been there at the beginning. I'm a little bit more cautious, and I say it's an extremely exciting idea. It's a scientifically reasonable idea. But again, the experiments that are done under controlled conditions in the laboratory are rather artificial. We buy the reagents that we use from a chemical company, and they come by mail. Well, at the origin of life, you're not allowed to send out for people to ship you reagents. It has to all happen spontaneously. On the other hand, to argue the other direction, you do have a huge amount of time, perhaps 100 million years, that to do these random experiments on the primitive Earth, and you can do them in 1 million locations at the same time. So now something that is perhaps seems rather unlikely, you multiply by 1 million times 1 million, and you can see how maybe it could have gotten started.
And more recently, RNA was right in the news everywhere was when the pandemic struck, because we had mRNA vaccines, Moderna, BioNTech everywhere. In India, we were the generic manufacturers once we got the chemical composition in place, and millions of doses were produced. One common, news everywhere was it appeared that it happened pretty much overnight. So how do you react to that? And people were, skeptical that no, I don't think enough research has gone in, etc., which probably wasn't the case. But you are in a better position to help us understand what exactly happened there.
Yes, Abhishek, I did think a lot of the concern and reluctance to get the vaccine, at least in our country, was fueled by the fact that it all happened so quickly. In fact, it all happened within 1 year. If you will recall, within the year 2020, at the beginning of the year, both BioNTech and Moderna started their projects, and by the end of the year, we had approved vaccines. And so how could this happen so quickly? Well, I'd like to think about this as a jigsaw puzzle that you have to put together, right? And so all of the pieces of the puzzle had already been discovered in the last decades before 2020. So we knew how messenger RNA worked. We knew how to make RNA in large quantities by copying DNA in the laboratory. We knew how to stabilize the mRNA so that it would not cause bad reactions in the human body. And all of these ideas were already there, and they were like the puzzle pieces laying out on the table. And what the vaccine makers needed to do was just fit them together. And that was important, and it was creative, and I'm not belittling what they did, but they never would have been able to do this if it hadn't been for the decades of research preceding 2020.
I think the Economist had mentioned that the RNA vaccines are a testament to the insight of Eddie Cantor, a comedian, that it takes 20 years to become an overnight success. So it's possibly that then it was all put together, and then it came to the fore when we all needed it. And yes, there was open source. All researchers who would not show their notes like you were in school, right? Kids would not show their notes to each other. That all went away. People who, researchers, scientists got together, shared each other's observations so that we could get there faster. How this science community, Tom, could you just help me understand? It's very competitive because, not everybody may want to show what's going on without making too much noise because it's your discovery. How does it work? And during such crisis, is it the human primal nature to collectively take on a force like the pandemic caused us to do that? Or is that very natural in the community?
Well, so you're absolutely right that the pandemic was a special situation, and everyone just naturally thought the right thing to do was to cooperate, to share, to help, to collaborate, to help everyone out. And there was really very little discussion about that. But otherwise, scientists are quite competitive, and they're sometimes careful about revealing everything that they know. I always encourage, whenever my students ask me if they're going to an international conference, they say, is it okay for me to talk about my experiments? I'm worried that someone might steal my ideas. And what I tell them is that if you are open about communicating your ideas, two things could happen. One is someone could steal your idea, and that's not good. The other thing is someone will help you go to the next step, and also they will remember the conversation. They will give you credit for the idea. And since you don't know which of these things has a greater weight to it, then do the right thing for science, which is to be open about your ideas and realize that occasionally you're going to be unhappy with what happens, but overall, that's the right thing to do. And
I think,
yeah.
Sorry, please go ahead.
I was just going to say, I think also the competition drives us to be better, right? Just as it does in the Olympics, just as it does in business, in your field, in commerce. If it weren't for competition, things would sink down to sort of a very low level of quality and creativity. And it's when you have to compete against other really smart, energetic people that you bring out the best in people. So competition overall is a good thing.
And this part about letting go and trusting science or trusting the other person to do the right thing, is that something you, for example, when you moved from quote unquote the DNA guy to RNA, that itself must have been a big change. How did you decide that, all right, I've got to be open enough, let's see what RNA has in store for me? And maybe you work all your life to do one thing and realize that the data is pointing you in another direction. How did you manage to let go? You would have learned new things there, but I'm talking about now a little philosophically in terms of life as well. It needed you to shed something, that unlearn something, and try to figure out what's happening on the other side. I wonder how you managed that.
Well, I have two comments. Thank you for that excellent question. First of all, the system we were trying to understand was so intriguing that we were so curious about how it could be working that it just sort of sucked us in, right? We just couldn't help ourselves. We had to understand. And the answer had to be in the RNA molecule, not in the parent of the DNA. It was the RNA that was doing these gymnastics, as we sometimes call them. And so that drew us into the RNA. And then the other thing I will say is that this was a small scientific discipline at the time. There were not enormous numbers of people working on RNA, but there were some leaders in this young field, and they took me in and were very supportive of me. And if we were doing something that wasn't quite up to, the standards, they would say, well, you're doing a good job, but you really should consider, learning this new technology. And then that was very helpful. So having the support of some key people in the community was also critical at that time.
And when you were on the path, you discovered something big in an experiment that you conducted yourself. If you could explain a bit about the tetrahymena experiment is what I read. And to quote you, and I find this funny, there is a video on YouTube posted around 13 years ago where you talk about your experience. And I quote you, you say, I did this experiment rather quietly. I didn't tell any of the graduate students what I was doing. I didn't want to look foolish if this reaction failed, as it was probably destined to.
Well, so yes, I was also teaching very heavily at the time, and so I had to only do experiments for myself at night, and often there was not very many people around. Yes, the tetrahymena, this is a pond animal. It is a single-celled organism which, like a human cell, has the DNA in the nucleus, and so the RNA is made in the cell nucleus and then shipped out into the cytoplasm of the cell. So it has some features of higher organisms, but it is as easy to grow and as cheap to grow as bacteria. So I thought that was an attractive system. So when I moved from MIT in Cambridge to Boulder, Colorado, I decided to set up my own laboratory studying this pond animal, tetrahymena. But there was another reason for being interested in it, and that is it had 10,000 identical copies of a particular gene. And that's a large number. Abhishek, most of your genes you have two copies of, one from your mom and one from your dad. So 10,000 is a big deal. And I was a chemist by training, and so having a lot of stuff to work with was attractive. And it turned out that the RNA made from these 10,000 genes was the one that had this catalytic activity, which we called then eventually, when we figured it out, we called it ribozyme, for ribonucleic acid with enzymatic activity. And it wasn't just me in the lab. Yes, I did this experiment late at night when nobody was watching, but I had some excellent people in my small laboratory, particularly Art Zog and Paula Grabowski, who I talk about in my book, who made key contributions as well.
Right. And how do you react when you discover something that isn't written in any textbook? None of the scientists are talking, something that is bizarre, first of its kind. How do you even process that? If you could flash back into, I think were you in your late 30s back in the day, or early 40s?
No, I was just barely 30.
Wow, amazing. Right. Okay.
I had just started out as an assistant professor. The first thought is that that it's wrong, that there's just a mistake in the experiment. And then so you repeat it a number of times, and when it's very robust, when it is reproducible, then you say, okay, it's not due to a mistake. But then we thought that it was due to a contamination, that there must be, because everyone knew, all of the textbooks said, all enzymes are proteins. And so clearly there was some enzymatic process taking place with this RNA. So okay, it has to be a protein. There must be a protein contaminating our RNA. So that was the first thought, is that we needed to work harder to get rid of this protein contamination.
Right. And I think one of your colleagues, when you guys were still figuring this out, gifted you a plastic daisy flower, didn't she? And I found that funny, where one of the petals said it's a protein. It's not, if I remember that
That's
correctly.
right, yes. And fortunately, we were able to think of something more scientific than picking the petals off the daisy flower to resolve this
Hmm.
problem. And it turned out it's not. There was not a protein involved. And this was the first example where RNA had been found to act as a catalyst in the absence of protein.
And there is this a line by Isaac Asimov. He says that the most exciting phrase to hear in science, one that heralds new discoveries, is not eureka, but that's funny. Is that something you agree with?
Yeah, yeah. Well, I think you get both of those happening. The
Yeah.
first, it's always the first one is always that's strange. That's funny.
Hmm.
That's the early one. And then the eureka comes when you figure out how the funny thing was happening.
And your case, it's in the family. You said your dad was a physician. Also, you've written about your wife. You married to a fellow biochemist. Does that also help? And she had a little hand in the experiment as well, is what you write.
Yes, she did. She gave us a key reagent at
Yeah.
one point that she had in her laboratory. And I also have two daughters who both became scientists. And I always wished that they would go into something else, like fashion or something like that, so that they wouldn't require advice from me. Sometimes the apple doesn't fall far from the tree. And so I am
Yes.
proud that the whole family has this scientific bent to it.
Lovely. And did you ever feel like screaming outside your balcony back in the day when you said, "Hey, RNA does far more things than you think it can, because I found something here," and it took the world a while to realize that, when you among your colleagues at the time were the first ones to get there, and after having reproduced those results, how did you try and deal with that bit? Did you ever feel like screaming into the pillow, for example?
Well, what was very important was that other people rather quickly found additional examples, right? And this is key in science, that you know, if only one laboratory can get an experiment to work, you
Hmm.
really worry that there's a problem there, right? But if other laboratories, including your competitors perhaps, get your experiment to work, this is a glorious day. And the
Hmm.
next example was with Sidney Altman at Yale University, working on a completely unrelated system to what we were working on. This was a bacterial enzyme that had both an RNA and a protein part. And he found that the RNA part by itself could be catalytic. And this was then a huge extension of our original finding and led to Altman sharing the Nobel Prize with me. And it did not take very long, Abhishek. It was
Hmm.
just 7 years after our publication, 6 years after Altman's publication, that we shared the Nobel Prize. So that's pretty quick.
Take me inside your lab, Tom. Back in the day, you have to do this cutting edge research. The first lab, you were the assistant professor, is when you got keys to it. It must have been a big day. How did the lab look like? If you could describe that, please.
It looked like something out of the 19th century. It had soapstone lab benches,
Hmm.
which had been used for many decades and were quite worn. It had oak wooden drawers. I think it had been used as a teaching laboratory. I think when we opened one of the drawers, we found the remains of a rat that had been dissected by a student. It did not have very good ventilation, but it
Oosh.
was mine, right? It was my own place. It was a place where my students and I could do what we wanted to do. And so to me, it was beautiful.
So there's a lesson in there. You don't have to wait for things to be perfect. Then you just
Yeah.
try and do what you're given, and hopefully you develop something at the end of the day which is beautiful.
Make the best of what you've what you've got. And now that we have beautiful modern facilities, I don't think the experiments we do are any better than what we did back then.
And the importance of repetition that you touched upon a minute ago, how we say even whether it's learning a musical instrument or trying to learn a new language or whatever, you make mistakes, you repeat, and hopefully you get there. In the kind of work that you do and your colleagues do, is that the key? And hoping that you don't lose your mind because you may not get what you're looking for, but you keep repeating it. Is that how it works then?
Yeah, I think that, repetition by itself does not get you very far, right? I mean, it
Ah,
might be it's a good ingredient,
hmm,
but I think that having a passion
hmm,
for your project is critical because science, if you're working on the cutting edge of science, it's always sort of two steps forward and one step back, or some weeks it's two steps back and no steps forward. And so you have to be really committed to wanting to understand the answer to something. And one thing I have found is that instead of telling a student what they should do in the laboratory, you want them to have some ownership of their own project. So you talk to them and you say, well, what would you like to do? You could work on this, or you could work on this, or you could work on this third thing. They're all of interest to me. You tell me what you want to do. And then if they choose what they want to do, and of course it has to be something that has to do with what we know about. It can't
hmm.
be astrophysics, that wouldn't make any sense. But as long as it has something to do with RNA and they articulate a reasonable project, I will say, good, that sounds good. Let's work on that. Let's see how you can make some progress on that.
And for those who are listening, and including me, until I read the book, this is painstaking work. To give an example from your book itself, one of your colleagues was transferring worms that were transparent from one petri dish to another, I guess, that were transparent and thinner than a human eyelash, and then the person was injecting tiny volumes of dissolved RNA in it. And you really need to have good eyesight and precision. I think threading the needle is easier than doing these things, and hope, and that is only the start of the project. You don't know what's at the end of it. So it's hard work.
It is hard work, and this, again, just like tetrahymena, has taught us much about what ended up being relevant to human biology. This transparent worm, roundworm, that is so tiny, has led to many discoveries that, again, turned out to be relevant to humans, but it was much easier to figure it out in this worm and then apply it to humans later.
And do you still get butterflies in your stomach when you do some experiments, if you still continue doing them? Or are you now a pro and it doesn't really happen that way?
I am now stuck in my office working on my computer, and I only get the joy of experimental discovery by talking to my students and my postdoctoral fellows, and they're the ones who are doing the actual laboratory work.
And what advice would you give them in terms of dealing with failure in the lab? Because young blood, 20s, 30s, and 40s, you might do something for years together, and failure is more like a cousin, perhaps, for lack of a better word. It's like Edison had said that I did not fail. What was it? 1,000 times I just was experimenting and then I got the light bulb out. So, what is your advice to them when you talk to your students?
So first of all, we try to we have this word called troubleshooting. It's a very long word, but trying to figure out why the experiment didn't work,
Hmm.
and then asking the student, well, that's the way science goes. Making them feel better about it. Everybody has problems, of course. That's too bad that you lost a whole week there, but that's the way it goes. Maybe it'll be better this week. But then also trying to look at it and say, is there something we can learn from what you did, even though it doesn't look very promising? Did we learn anything? And then sometimes you say, oh yeah, it looks like, maybe this part of the experiment might have worked. And then I say, well, okay, we can't believe that yet, but maybe it did. So why don't you now do a new experiment focused on that part that you think worked and see if it really is working, right? So I don't know. It's just like all human endeavors, right? You have to learn to deal with failure and to celebrate success.
Right. And can a student who's got lower grades in a classroom still ace it in the lab? Or is there a correlation there?
Very little correlation. And I was always very good in the classroom, always getting the top grades. And I'd say my laboratory skills were rather average. But I had a student who was very poor in the classroom, had to take exams over and over again, and was so creative in the laboratory. And he's had a wonderful scientific career, is quite famous, and has been elected to the U.S. National Academy of Sciences. And so clearly the correlation between success in the classroom and success in research is fairly random.
Right. And what is not random is the folks who go to Stockholm and are feted with the highest prize in science. Tell me, Tom, where were you when you got that phone call that you have been elected to receive the Nobel?
Well, I was at Harvard getting an award, and I was sharing this award with a good friend, Joan Stites, who is an RNA scientist at Yale and was one of the ones who was helpful to me when I was starting out. And so Joan Stites and I had just received the Warren Triennial Prize given every 3 years at Harvard. And during the dinner, they said that many of their recipients got the Nobel Prize within 10 years. Well, about 7 hours later, in my hotel room, the telephone rang, and it was Stockholm, and they told me that that Sidney Altman and I were sharing the Nobel Prize in chemistry in that day in 1989. So I was completely unexpecting this. I was very shocked. I was very nervous, and I put down the phone, and it rang again, and it was David Baltimore, a Nobel laureate from MIT. And he said, I just heard the news, Tom. He said, come on over to the Whitehead Institute, and we will take care of all of the press, all the questions and everything, and you can have a good time with your friends here at MIT. So that was a wonderful day that I had, because I had been a student at MIT, and so it was great to be at MIT and Harvard when I got the prize, and then I flew back that night and had more celebrations back in Colorado.
Until you got that second phone call, did you suspect that the first one was a crank call at all? Did you?
Well, the first call, they put on a friend of mine from Stockholm, and just to tell me in person that this was not a crank call. But the truth is that because the phone was ringing with all kinds of reporters from all the newspapers, it would have had to have been quite an elaborate joke.
Yeah. Yeah. And what was it to accept that prize on the day? If I were the audience, what would I be watching, and how did you do on the day?
Well, it's not a day, it's a week. They organize dinners and concerts, and you go to the American Embassy, and you go to the various lectures, and it's really quite an elaborate week. And my family, and I also invited the people from my laboratory and their families, joined me there in Stockholm. And for many of them, it was the greatest week of their life. For me, I think I was a little too much in the spotlight to really feel very relaxed about it, but it was certainly the most memorable week of my life.
And I think a few books have acknowledged that just fell down from the library up there. You write somewhere that you forgot to take a bow. Was it in front of one or two bows is what you forgot in front of the king and the queen, and then to the Royal Academy, and then to the audience or some such? Perhaps the spotlight caused you to do that.
Yeah, yes, they had given us a dress rehearsal on the stage where we practice bowing and the order of the bows to the king, to the Royal Academy and to the audience, and I screwed it up I did okay during the practice. I
Right.
only screwed it up for the real time, but it didn't matter, right?
Absolutely not.
Gave me the medal, even so.
Yes, and did that change your life in the weeks leading after the big day? It must have been quite a buzz as a young, you're barely 41 or 42 at the time, weren't you? If I
Yeah.
got my calculation right. So I am going to turn 42 next year. So I can only imagine what it might be for someone that age to get there. How was it? How did your neighbors, your family, your parents, colleagues, etc., treat you uh, after that?
So, I think that in the scientific community, I already had a good reputation, and so not much changed. But
Hmm.
in the but what changed was in the city, in the when I would go into the grocery store or to the school,
Yeah.
and previously it had a wonderful anonymity, and now all of a sudden I was known and I was recognized. And this took place this continued for quite a few years. And then, fortunately, about a decade later, some more Nobel Prizes came to our university in the physics department. And then the pressure was off of me.
Right. That Robin Williams, once upon I'm not comparing the Oscars and Nobel at all, but he'd been asked, how did it feel to win the Oscar for Good Will Hunting? He'd done a brilliant job there. He said, in the first week, they said, congratulations, great stuff. Second week, yeah, way to go, Good Will Hunting. Third week, hi, Mock. Because he was there as Mork and Mindy, one of those sitcoms back in the day when I wasn't even born. But people, slowly got to their usual lives. Obviously, for you, it wasn't that. You still had
It
to
took
wait
a
for
little longer.
it took a
And,
little.
and by the way, that Mork and Mindy show took place in Boulder, Colorado.
Oh, really? What are the odds?
The, the
Brilliant.
house, the house is still here.
Lovely. And one last one is this is a bit of a cliched question, Tom, but with the benefit of the wisdom that you've amassed over the years, what advice might you give yourself who was 21? If you were to tell something to your 21-year-old self or 25-year-old self with the benefit of hindsight.
So my 21-year-old self. So this was when I was still in college and or maybe about ready to graduate from college. I think this may sound strange, but I probably studied too hard. I was in the library doing homework all of the time and reading books, and that's important. But I think it's also important to talk to people, to learn from other people. I was rather shy. I did not have a lot of friends. I was fortunate to have a girlfriend, and that was very important, who became my wife and is still my wife. But I think that I would encourage people to have a full life and to work hard, but also to play hard.
Lovely. And this is the book that I'm holding up for those who are not listening on Spotify or elsewhere, but on YouTube. And one thing that reached out to me, Tom, is that it speaks to the common man. And it must have been a bit of a challenge for you to leave out. How did you decide how much to leave out? That must have been quite difficult, I'd imagine.
Abhishek, I'm so excited that you found it readable because that was my goal. And I will tell you that every time I wrote a chapter and sent it to my editor, she would say, it's too technical. You need to make it simpler. And I would work harder on it and send it back. And eventually it got to a point where she thought it was okay. But it is difficult for a practicing scientist to leave aside the jargon that we use in the laboratory. Every field has
Right.
jargon, right? It's a good thing in the laboratory. It makes it easy to have a conversation very quickly. But when you're trying to extend your reach to the non-scientific public, it's a challenge. And I did work very hard at it, and I thank you for saying that I was at least partially successful.
Absolutely, Tom. Thank you very much for the book as well as for your gracious time on this podcast. Thank you so much. I really had fun talking to you, Tom.
Likewise, take care.
Thank you.